Cellular respiration occurs in all living organisms and is characterized by the creation of ATP as well as reduced electron carriers. These carriers undergo a series of redox reactions to generate a proton motive force. The potential energy of this proton motive force is then harnessed by a membrane known as ATP synthase in order to generate ATP, which the cell can use to carry out its various functions.
Cellular respiration can be broken down into three distinct steps: glycolysis, the Krebs cycle, and the electron transport chain. While glycolysis and the Krebs cycle produce some ATP, their primary function is to catalyze the electron transport chain by producing reduced electron carriers.
The most common reduced electron carrier used in cellular respiration are NADH and FADH, both of which can donate an electron to the electron transport chain in order to begin generation of the proton gradient. This proton gradient is established across the intermembrane of eukaryotic mitochondria or across the cell membrane of prokaryotes.
At the start of the electron transport chain, complex 1 receives 2 electrons from NADH, generating NAD+. Complex 1 then passes the electrons to an intermembrane protein and pumps 4 protons out of the matrix and into the intermembrane space.
The electrons picked up by this intermembrane protein undergo a series of redox reactions with other transmembrane proteins, which increase the proton gradient. At the end of the electron transport chain, electrons are passed to molecular oxygen, which becomes reduced to water, ultimately pumping more protons into the intermembrane space. This potential from the gradient is ultimately used to power ATP synthase and drive the formation of ATP.
ATP synthase is made up of 2 sections. The first portion is similar to a flagellar motor and acts as a transmembrane proton channel. The second is similar to DNA helicase. They work together to create a molecular turbine, shuttling protons across the membrane. The spinning motion created by this turbine binds ADP and inorganic phosphate, creating 3 ATP every time the motor turns.
The method described is more formally known as aerobic respiration, and while this is the most commonly used method of respiration, there are variations to this process which still allow for the creation of ATP.
The most notable deviation from aerobic respiration is known as anaerobic respiration, and is highlighted by the fact that it substitutes other molecules in the place of molecular oxygen to act as the electron transport chain's terminal electron acceptor.
The most common substitute for molecular oxygen is hydrogen sulfide, which can also pick up electrons and allows the electron transport chain to continue propagation. The biggest difference between using molecular oxygen and hydrogen sulfide lies in their respective redox potentials. The greater the redox potential of a terminal electron acceptor, the more energy released during respiration, and thus the more ATP one can generate.
The earliest cells lived in anoxic conditions, an environment void of oxygen. As a result, the cells used terminal electron acceptors other than oxygen, which were abundant in their habitats, to perform cellular respiration.
Once cyanobacteria started the Great Oxygenation event, some bacteria adapted, allowing their electron transport chains to utilize oxygen as a terminal electron acceptor. The bacteria that utilized oxygen saw higher energy yields, giving them a giant selective advantage over bacteria that used other electron acceptors.
This advantage allowed the oxygen-utilizing organisms to reproduce at higher rates than the anaerobically respiring organisms, and have higher functioning metabolisms, ultimately leading to a dramatic shift in the preference of respiring organisms.